Hello, everyone. Today we are going to study a very important concept, yet it's a very confusing concept, which is the effect of the cholesterol molecule on the fluidity of the cell membrane. We all know that the cell membrane consists of a phospholipid bilayer. As you can see here in the diagram, this is the phospholipid bilayer. A phospholipid bilayer is seen under the microscope as what makes the cell membrane. So what is a phospholipid? It's a molecule that consists of a phosphate head, as you know from your study of the cell, and two lipid tails. And the two lipid tails are made essentially of fatty acids. And as you know from your study of biochemistry, that those fatty acids can be saturated fatty acids, meaning that all the molecules inside them are a single bond, all the bonds in between the carbon atoms are single bonds, as we will see on the next diagram, or they might be unsaturated fatty acids with some double bonds present in the molecule. Since we have two layers of phospholipids in the cell membrane, it's called a phospholipid bilayer. As you also know from your previous study that the phosphate heads carry a negative charge, and that's why the phosphate heads are said to be polar, and they are hydrophilic, meaning that they love water. And if you imagine that this is a part of the cell membrane, then you will have the cytosol inside the cell membrane, which is mainly made of water or which has a good deal of water. And you have the extracellular fluid outside the cell that also contains plenty of water. That's why even if you mix up those phospholipid molecules, you will find that they will automatically rearrange themselves so that the polar hydrophilic phosphate heads are always directed towards water, whereas the lipid tails or the fatty acid tails always hide inside because they are nonpolar. They do not carry any charge, whether a true charge or a partial charge. And they are, of course, as you know, since they are non-polar, then they are hydrophobic. They fear water and they tend to clump away from water. This is the structure of the cell membrane or the phospholipid bilayer. As you might have heard before, um, the structure of the cell membrane is described as being a fluid mosaic structure. Fluid because if you leave those two phospholipid layers on their own, they would slide over each other. They would shift laterally over each other. That's why in between the phospholipid molecules, we have several protein molecules that give the phospholipid bilayer a mosaic pattern. They are not seen here on the slide, but they lie in between the molecules, the phospholipid molecules. They might be integral proteins, like filling the whole thickness or traversing the whole thickness of the cell membrane, or they might be peripheral protein present only on the surface of the cell membrane. Another type of molecule, the protein molecules I've just talked about give the mosaic pattern to the cell membrane. But there are other molecules that are inserted in between the phospholipid molecules that are not protein in nature, but that are lipid in nature, or more specifically speaking, they are steroids, as you might have studied in your previous biochemistry session. And those cholesterol molecules also lie, in addition to the protein molecules, in between the phospholipid molecules. In the past, we used to say that cholesterol diminishes the fluidity of the cell membrane and helps diminishing this side to side sliding of the layers of the phospholipids over each other. But recently, it has been proved that cholesterol has different, even opposing functions depending on the temperature. But let's first, before studying this and studying the effect of cholesterol on the fluidity of the cell membrane, let's first see the structure of the fatty acids and the structure of cholesterol. So our video today, is talking about the effect of cholesterol on the fluidity of the cell membrane. Does it only increase the fluidity? Does it only diminish the fluidity? Or does it do both? The goal of this video is to prove that this depends upon the temperatures or the ambient temperature. So in cases of hot weather and high temperature, when the membrane tends to be too fluid, and we will know why, cholesterol helps diminishing the fluidity and providing more rigidity to the cell membrane. On the other hand, if the temperature, the ambient surrounding temperature is low, the molecules of the cell of the phospholipid bilayer, especially the lipid tails, tend to clump together and to be too rigid. The cell membrane is likely to be too rigid, and in such a case, cholesterol presence in between would help diminish rigidity and increase fluidity. So they are two antagonistic actions, opposing actions, but in both cases, they help maintain like an average amount of fluidity or rigidity in the cell membrane that allows the cell membrane to carry out its function. Because if the cell membrane is too rigid, then it would probably interfere with the passage of substances across the cell membrane. And if it is too fluid, then there would be difficulty in maintaining the shape of the cell, and therefore, again, it would be difficult for the cell to carry out its function. As we said before, the lipid tails that are present in the phospholipid molecule are nothing but fatty acids. What are fatty acids? They are organic molecules. Actually, they are hydrocarbons, meaning that they consist of carbon atoms, the ones you see white here, and hydrogen atoms, the ones you see blue in color. And those, or this long hydrocarbon molecule ends by a carboxylic group, C double bond O, OH. The red atoms are oxygen atoms. The presence of COOH at the end of the hydrocarbon molecule, and this COOH is called a carboxylic group. It is a functional group which infers acidity to the molecule, and therefore they are called fatty acids. If you have no double bonds between the carbon atoms at all, until here, we do not consider the carboxylic group. If in the hydrocarbon chain you have no double bonds at all, this is because every carbon atom in this hydrocarbon chain is surrounded or has 4 single bonds. So it is saturated with hydrogen atoms. There is no missing, or there are no missing hydrogen atoms. So saturated fatty acids means there is no double bond and the molecule is saturated with hydrogen. But if If we come here, for example, if you look at that double bond, there is a double bond, and that double bond is here. Why? Because we have removed this hydrogen atom and that hydrogen atom by dehydrogenation. When you remove the two hydrogen atoms, then every carbon atom now needs to form a double bond with the neighboring carbon atom, so that every carbon atom is again surrounded by four bonds, one, two, in addition to the two in the double bond, because as you know, carbon is a tetravalent element. So here we have only one double bond, so it is an unsaturated fatty acid. You might have two, you might have three double bonds, and the more double bonds you have, this simply means fewer hydrogen atoms that are allowed, and this means that the hydrocarbon chain is not saturated with hydrogen, and that's why we call it unsaturated. If the two hydrogen if you look here and there, both contain a double bond. So both molecules represent unsaturated fatty acids. But here you can see that the hydrogen atoms are present on opposite sides of the double bond, whereas here they are on the same side. If they are on opposite sides, then we call this a trans compound. Trans means on both sides. If they are on the same side, we call this a cis compound. The main difference between saturated and unsaturated fatty acids is not only in the presence or the absence of double bonds, but it's also in the shape of the molecule. Here you can see this is the overall shape of the molecule. You have a carboxylic group, and or if this diagram represents a phospholipid, but I don't think so because we have only one lipid tail, then if this is the saturated fatty acid, this is the carboxylic group, then you find that the hydrocarbon chain is straight. But in case of the unsaturated fatty acid, you find that it is kinked. Why? Because the double bond is rigid. And because the double bond is rigid, so when there is a kink in the molecule, this kink cannot be easily moved. So if you have plenty of saturated fatty acids, one above the other, because they are all linear, then they would be parallel to each other, and there would be minimal spaces in between them. But if you have kinked molecules like those of the unsaturated fatty acids, whether trans or cis, then they will not be parallel to each other. They would have wide spaces in between each other because of the rigid kinks, and that's why the more you have of the unsaturated fatty acids that have double bonds, the more fluid the fatty acid solution or the fatty acid substance is, because it contains wider spaces, wider intermolecular spaces. This was important to know before going deeper into the subject. Furthermore, about the cholesterol molecule, the cholesterol molecule is one of the examples of steroids, because in the middle it has a rigid steroid nucleus that is fixed in all steroids, whether you are talking about cholesterol or about cortisols or about sex hormones. You have this rigid steroid nucleus, and it is rigid again because it has double bonds. And this steroid nucleus is a four-ring nucleus, four rings that are fused with each other. So it consists of cyclic hydrocarbons, and it is nonpolar because it does not carry any charge. In addition to this steroid central nucleus, we have a lipid tail that is also made of hydrogen and carbon, and that is nonpolar. So both the rigid central steroid nucleus and the lipid tail are hydrophobic, The only hydrophilic part is the hydroxyl group, because this hydroxyl group can lose a hydrogen atom in or a proton in water, and therefore it would carry the oxygen would carry a negative charge. These, of course, are two different isomers of cholesterol. How does cholesterol arrange itself in the cell membrane? As you might have studied in your biochemistry, it arranges itself in such a way so that the polar hydroxyl group that you see here in red will interact with the phosphate heads of the cell membrane, because polar likes polar, whereas the rest of the molecule, which is the central hydrophobic steroid nucleus and the hydrophobic tail, will be in the central part away from water, interacting with the lipid tail. In such a case, it's obvious that cholesterol, by being present in between the phospholipid molecules, will interact with them and will affect their motion. And we will see what happens in case of high temperature and what happens in case of low temperature. In case of high temperatures, and we will see why later on, the unsaturated fatty acids that tend to be liquid increase, and the membrane starts to be very fluid, to the extent that if it is left like this, it would interfere with the function of the cell membrane. So the intrusion of the cholesterol molecules in between, in such a case, and in such a pattern, would diminish the fluidity, would diminish sliding of the two phospholipid layers one over the other, and therefore will increase the rigidity of the membrane. How is this sensed by the cell? How is this done? This is what we are going to see on the next slides. When the weather is cold, the lipid tails tend to clump and tend to be solid. Why? Because the cell membrane has a particular way, using certain enzymes, to convert many of the unsaturated fatty acids that make the cell membrane fluid, to convert them into saturated fatty acids. And as you know, the saturated fatty acids tend to be solid, especially at low temperatures, just like the margarine, for example, or the hydrogenated fatty oils become solid in the fridge or in cold temperature. In such a case, the presence of cholesterol in between the lipid tails, we cannot say here that it will diminish the fluidity and increase the rigidity. On the other hand, its presence would be beneficial, because since the lipid tails start to clump and tend to clump together, the presence of a foreign molecule in between diminishes clumping and increases fluidity. So this is what we're going to wrap up on the following slides. So what happens actually is that there is a change in the phospholipid fatty acid composition to affect membrane fluidity. In colder temperatures, the cell membranes are keen on having a higher percentage of saturated fatty acids. I apologize. In colder temperatures, the cell membranes, if they stay having higher percentages of saturated fatty acids, then they will become very rigid and they will become less fluid. And this definitely, as we said before, will negatively affect cell structure and function. In such a case, if the cell membrane stays having or preserving this high percentage of saturated fatty acids, then cold weather will be a real challenge to the cell, because it will be a real challenge to the fluidity of the cell membrane. So the cells have to find a solution. They should try to enhance their membrane fluidity in cold weather. How can they do this? They think that modifying their structure, incorporating more unsaturated fatty acids with more double bonds, more kinking, and larger spaces in between the molecules would help fluidity. So the solution when the cell membrane is exposed to cold weather is that it has to find a way to convert some of its saturated linear fatty acids into unsaturated kinked fatty acids to increase the spaces in between them and increase their fluidity. So as we said before, if the membranes stay having or remain with this high percentage of saturated linear fatty acids, they will become very tightly packed, they will become less fluid and very rigid, and therefore they might appear as if they were frozen, and this would hinder the transport mechanisms across the cell membrane. So what are the proposed solutions? The first solution is from the part of the cell membrane, but the second solution will be from the part of cholesterol. From the part of the cell membrane, when the membrane is in cold weather, it will try to convert some of its saturated fatty acids into unsaturated fatty acids using an enzyme called desaturase enzyme. As its name indicates, the desaturase enzyme helps converting the saturated into desaturated or unsaturated fatty acids. So the desaturase enzyme will be formed, and the gene for the desaturase enzyme formation will be turned on only when the weather is cold. The other solution is the role played by cholesterol. As we have said before, cholesterol has a central, rigid, hydrophobic, nonpolar steroid molecule, and this hydrophobic steroid nucleus is towards the hydrophobic interior, towards the lipid tails. And meanwhile, it has two hydrophilic polar ends, or actually it's one hydrophilic polar end, that is the hydroxyl group. So one part is hydrophobic, the other is hydrophilic. So it is an amphipathic molecule. So this cholesterol molecule will interact with the cell membrane, as we have explained before, and will result in a diminution of the rigidity and increased fluidity by preventing clumping of the saturated fatty acids. On the other hand, when the temperature is high and the membrane tends to be very fluid, the mere presence of cholesterol molecule intruded in between the phospholipid molecules will restrict the movement and restrict sliding over of the layers one over the other. So at lower temperatures, cholesterol will tend to increase the membrane fluidity, whereas at higher temperatures, when the membrane tends to be very fluid, cholesterol tends to restrict the membrane fluidity. This process of adaptation using desaturase enzyme and cholesterol occurs in the freshwater organism E. coli. And this is a bacterium that lives in polluted fresh water. And this is a kind of adaptation to changes in the environmental temperature, and in particular in cold weather, because if this adaptation does not occur in cold weather, the cell membrane and the cell wall, the cell membrane in particular, would be frozen. So this provides the E. coli bacteria with more resistance to cold. As we said before, the presence of the changes in temperature result in activation or inactivation of the desaturase enzyme, and this activation and inactivation is not only by activating the expression and or inactivating the expression of the gene, but also because the temperature changes certain bonds inside the enzyme. And therefore, when the bonds, as you know, are changed, the conformation or the 3D configuration of the enzyme changes. So temperature changes induce bond changes and conformational changes in the desaturase enzyme that is capable of changing the number of double bond in the fatty acids. So actually, the enzyme senses or feels the temperature changes just because these temperature changes affect the enzyme itself, affect its internal bond because the enzyme is a protein, and therefore affect its conformational change and its activity. Because, you know, when the enzyme changes its 3D shape, it changes its active site, and therefore this change might be favorable to bind with the substrate or unfavorable to bind with the substrate. I hope this video was useful, and thank you so much for having watched the video.